Hello, my name is Arvind Krishnan, and I'm a rising fourth year at the University of Chicago, studying molecular engineering and biology. Today, I'll be talking about my work this past summer in the Olopade Lab, which is focused on understanding breast cancer, specifically in patients of African ancestry. Breast cancer is a very heterogeneous disease. As such, it's important to classify these tumors into various subtypes in order to find the best treatment for them. Moreover, an emerging field in cancer research is immunotherapy. That is, how can we use the immune system to fight cancer? Therefore, it's important to understand the tumor microenvironment in order to see how effective these immunotherapies can be. Therefore, the steps involved in our pipeline are, first, using NFCore to perform RNA-seq alignment with the reference genome. Then using the expression profile it generates, we're able to perform cell type deconvolution. That is, seeing the different immune cells inside our tumor samples. Following that, we look for various batch effects in the expression profile. And after that, we use PAM50 to find the subtypes of our tumors. The dataset I used to develop this pipeline is composed of RNA-seq samples from tumors taken from patients at UChicago Medicine from 1997 to 2019. The first step of my pipeline was to use the NFCore RNA-seq analysis tool. What this does is it reads the raw messenger RNA sequencing data from our tumor samples in the form of FASTQ files. Then after trimming the adapters used for sequences, it aligns the RNA segments to a reference genome. By doing that, we can quantify how the RNA segments align to the reference genome and generate an expression profile. NFCore also allows us to perform quality control. We can check for issues like sequencing errors, overrepresented sequences, high duplication levels, and more. For example, here we see that the sequence quality is high across all the base pairs read. The next part of my pipeline was to perform cell type deconvolution. Using the expression profile generated by the NFCore tool, we used a variety of methods to estimate the immune cell content within a sample. The different methods quantify the different immune cell types differently, but the way they generally work is by using a system of equations that describe the gene expression of a sample as the weighted sum of contributions of the various cell types. Our analysis allowed us to look at the cell type diversity within a specific sample, as can be seen on the right, as well as compare the prevalence of a particular immune cell phenotype across the different samples, as you can see on the left. At this point, we look for batch effects in our data. When conducting an experiment, the variance in our results can be attributed to biological factors, such as the mRNA expression levels in our data that we're interested in, as well as technical factors, such as the instrument name we use to sequence our RNA. Therefore, in order to remove technical variation from our data, we look at the expression levels within and across the various batches. Here, we're evaluating the effects of the sequencing machines used as indicated by the different colors on the X and Y axis. We see that the variables are not linked to any particular clusters in its expression. Next, we move on to classify our tumor subtypes. To do this, we use the PAM50 method developed by Peru et al. And what this method does is it uses 50 particular genes from our dataset, and using that, it puts the various breast cancer tumors into the various subtypes. These 5 different subtypes are determined by the various receptors within the tumors and that vary in their prognosis. These 5 different subtypes are determined by the various receptors within the tumors and they vary in their prognosis. Here's the distribution of the various subtypes in our samples. I hope you enjoyed hearing about my project over this last summer. The scripts I used to create the pipeline are reproducible and are available on my GitHub. Thank you.